Topoisomerase cloning is based in the action of the enzyme DNA topoisomerase 1, which clips DNA and rejoins it during replication. Topoisomerase 1 has restriction enzymes and ligase activity to enable the DNA to unwind. One of the enzymes most widely used is topoisomerase 1 from Vaccinia virus. The enzyme clips in only DNA strands, freeing a thymine, and it stays attached to DNA by forming a covalent bond with a phosphate group attached to the thymine. After relegating the ends of the cleaved strand, topoisomerase 1 releases itself from the DNA. The activity of the enzymes can be used to clone PCR products since the topo vectors and provided linearized with topoisomerase 1 covalently bound to each 3-phosphate and ready to ligate PCR products with compatible ends. PCR 2.1 topo vector is used to insert PCR products, simplifying the traditional restriction and ligation cloning with one-step cloning strategy and limits the use of any enzymatic modification of the PCR products and does not require the use of primers. The vector is linearized with 3-thymidine overhangs for direct ligation of Taq polymerase, amplifying PCR products. And it is activated with covalently bound topoisomerase. For cloning and transforming this vector into chemically competent cells, it is necessary the addition of sodium chloride and magnesium chloride to the ligation restriction that increase the number of colonies over time. For cloning and transforming into the electrocompetent cells, cell concentration must be reduced in order to prevent arsenate. For this, it is necessary to make a dilution for achieve the proper salt concentration. The selection method to verify the presence of the insert that is mostly used is black-white screening. This method uses a mutated bacterial strain and a medium containing X-gal to produce white or blue colonies. Another method can be the use of restriction enzymes because the plasmid contains recognition sites for restriction enzymes in the place where the PCR product should be. The plasmid can be extracted and cut with these restriction enzymes, and the results are shown in an electrophoresis hall. The plasmid also contains fusion proteins that reports if the plasmid is inserted into the cells or not.
This protocol consists of 8. The first step is to unfroze the PCR product. Then you unfroze the reactives of the kit of the PCR 2.1 topo plasmid and you keep them immersed. Then you do the ligation reaction for the chemical transformation or electroporation transformation. Then you incubate it at ambient temperature for 10 minutes. Then you transform 3 microliters of ligation in 15 microliters of chemiocompetent cells and electrocompetent cells. Optionally, you can keep the ligation at -20 for its after use. After transforming, you add 300 microliters of stock medium and you incubate it at 37 degrees. After that, you spatulate hold the volume in a LB plate with ampicillin and X-gal. You keep it incubated to 37 degrees during all night. Next day, you take a picture of the plate and you keep it in the fridge. These colonies will be used in the next practice for the extraction of plasmid. For that, it's recommended to grow 2 colonies, one white and one blue, of the ones that were grown in the LB plate with X-gal and ampicillin.
Interrupting the fragment of the gene that codes for the beta galactosidase them after 15 hours instead of 24 hours as stated by the protocol. It could also be that the competent cells were stored for a very long time and they decreased its viability. It could also have been that the DNA was not from the best quality possible and this lowered the efficiency, or that the plasmid